Neurodegenerative diseases are some of the most studied conditions in the biomedical sciences. But are we making sense of the vast streams of data collected by scientists? How do we make sure of its quality, or make sure other researchers can find it so that information isn't wasted? Well, my guest today curates this data with a fine-tooth comb. She is a biomedical data manager at Sage Bionetworks. See how someone brings creativity to bioinformatics in a way that can accelerate a new era of innovation in Alzheimer's disease. This is the Discovery Art podcast. I am Drew Douglin, and now let us welcome Trish Zintel. Trish, rumor has it that you started out your career as an illustrator. So tell me about that background before growing up. What is that story, and when did you realize that you had that artistic flair?
It's a long story that I am forcing myself to try to skip over, so we're not here forever. But my earliest memories of getting into art were actually through media. So my first foray into art was kind of recreating art I couldn't afford, as it was related to things like music or movies, and just to be completely cringe. My first big obsession, music-wise, that was my own and didn't come from my dad, was Linkin Park. And I got so excited because when I got into them, I also found out that at least two of the members, if memory serves, have art degrees, and it made me really excited. So Mike Shinoda went to an art college in Pasadena. I don't remember which one. And then I think Johan also as well. And so I was kind of just like recreating what I saw in the things that I really loved about their band's artwork, which I then came to find was made by artists and not just like Microsoft Word art. But I was like 12 maybe, and just plastering it all over my walls. And then my dad showed me the movie The Crow, which also has a phenomenal soundtrack, but very emo and moody. And so some of the first like drawings I remember actually putting together with charcoal, which remains to be my favorite medium, were some portraits of Brendan Lee playing the character, and like this silhouette of him standing in the window, and then it kind of like ran from there.
Linkin Park was my first ever CD I bought, so also
Me
big
too!
fan. It must be the same kind of time.
My dad was thrilled that I finally chose rock music instead of Britney Spears, because I'd forced him to take me to that concert and he wasn't technically thrilled by it.
Was there an intersection of art and science that you enjoyed at the time, or did the interest in science sort of come later?
Science kind of always made sense to me without really thinking about it, at least until I got to undergrad. So I didn't put a ton of effort into science, and I spent a lot of my effort on my art instead. But I had always been pretty interested in both. And so the intersection came when I realized, holy crap, college is expensive, and I am a first-generation college student, and I had to sign loans to go to college at the age of 17. And it was terrifying. And so I was like, well, I guess I didn't make the decision while I was signing student loans, but when I was trying to find where to apply, I happened upon scientific illustration. And at the time, I remember only being able to think I could apply to Rochester Institute of Technology, Savannah College of Art and Design, and Arcadia University, which is where I ended up going. And yeah, it was my attempt to still be an artist and not to have a livelihood afterwards, by making it science-based as well.
What did your family think of that decision? Do they support the artist track, or were they happy that you sort of combined it with sort of this scientific technical expertise?
They were always really supportive, if cautious. So they briefly, they had to file for bankruptcy because my mother had an autoimmune disorder that doctors told her she had Munchausen's for like more than 2 years and neglected to diagnose. So that's part of the science piece in my upbringing. But they could, long story short, they could not co-sign any of my loans. My grandmother had to. Um, and so they were like, well, I mean, we aren't even paying for any of this, so it would be beyond us to tell you what to do. We just really hope you pick something that you're going to be able to afford to pay these back. And so actually the switch from art major to biology major happened when I realized, naively probably, that that I didn't like the pretentiousness of some of the art conversations that were happening in some of my first year art classes that I think I could absolutely handle now, but I was like, no, and like ran away from it in my first year of college. And then I really liked the biology piece, so it kind of just shifted that way. And I also found out that I would need at least a master's degree in scientific illustration to really make a career out of it. And so my first instinct was, no, I couldn't possibly take out more loans. So I need to go find a career over in biology instead.
Do you feel like then when you made that switch, the background in illustration helped? What was that like?
Yeah, it did help. And I don't know if I was necessarily very good at explaining why, but my version of studying was just doing what I did with art, which was rewriting and recreating the information that was presented to me in a way that made sense for my brain, and then tutoring other students. And so it still felt like art because it was still creative for me. And I am one of, I don't know how rare it is, but I need like a lot of information. Like I need to be given a ton of information and then to like condense it and make it kind of work within that framework for me to fully understand it. And that's how I prefer understanding things. And so, yeah, it, to me, I guess the wires already had been crossed in my brain, in terms of just using my my artistic and creative skills in the context of biology. But it also really helped me be a good tutor. It ended up being a really great way for me to be a science communicator, which I got into more in grad school. And then one of my favorite things to do in grad school was teach and make slides to present information to students. And so it ended up kind of all fitting together. In retrospect, I don't think I was that cognizant of it at the time.
Yeah, so not only did you transition to science, but then you embarked on the major journey in science, which is a PhD. So
Yeah.
what sort of brought you to that decision? You know, that's a big career choice. Did you think you were going ahead into research as a career at the time?
I had no idea that research was even an option until probably like my third or fourth class in my intro biology class, where the person who would become my first academic advisor in biology, Dr. Lauren Howard, said, hey, we take on research students in our lab. And I went, wait, you can do research as a career? And my mind was blown. And so that is how I got into research for the first time in a dendrochronology lab.
What is that?
Dendro, I think, is the root for tree, and chronology is time. And so it's studying environmental impacts of a region based off of the signs they leave within tree ring growth. So it's a lot of like taking, it's a very like lumberjacky type ecology, like research field. And so, yeah, like it was a lot of like using power tools to sand down um, uh, tree cross samples, and then working with them to date them, and then looking for fire scars um, within that research to try to figure out what the ecological history of, I think it was a forest in West Virginia, or it could have been the Pine Barrens in New Jersey. I'm not sure. And yeah, and so trying to study it that way. It was, I eventually, my fear of power tools overpowered my interest in that subject, and I discovered how much I loved genetics. So then I pivoted to a developmental genetics lab, and worked with fruit flies.
Wow. That is quite a transition. And yeah, absolutely bummer if you're a lumberjack who doesn't like the power tools.
My dad was so excited because it was the only time I went home for college during the summer, and I brought like big tupper, like big bins worth of tree samples, and he got to teach me how to use the power sander. So my dad was so excited.
He's like,
and then
yes.
yeah, and then he was like, and I was like, I don't think this is for me. And he was like, crap. Okay, all right. Let her go that way, but it was nice for us to bond over it. And then I met my second academic advisor, Cheryl Smith, Dr. Cheryl Smith, and she's still an incredible friend to this day. So she was really the reason I even found out that a PhD was possible, and that I didn't have to pay for it in the traditional sense that you would in terms of taking out loans before you're there. That I could get a stipend to do it, and she helped me get there.
How was your PhD? I mean, it can be a really tough time. How did you find it?
Oh, it was rough because for a lot of reasons, I have a fairly young family, and so the first time I had lost anyone was during grad school, like some grandparents and stuff like that, and then I was also doing cell culture, so it wasn't something I could just run away and leave necessarily for things like that. I didn't I didn't know how to form the right kind of mentoring network, and I relied entirely on my advisor, who didn't have the space to give me what I needed. And so it was a lot of teaching myself how to do things, and that meant a lot of external stress from the program itself and a lot of internal stress. And so I didn't really know how to take care of myself then, and that made it pretty difficult. But I can't say that I regret doing it. So
Yeah, and yet you were still driven to pursue postdoc research.
yeah, I vehemently didn't want to. But I knew that I hadn't had enough. So I guess for some context that feels too much like bragging, but I'm going to say it anyway. I finished my PhD in 2019 after 5 years of grad school, and by the time I was 27 years old. And so I did it pretty quickly. And a lot of that came from this stress of knowing that I had all of this debt coming my way that I was going to have to start paying back, and I needed to be able to do that without like completely fumbling, because not only was my debt student loan debt, most of it is from private student loans, and so that carries a much higher emotional burden, at the very least, for me than my federal student loans, which do exist. And so I knew that a postdoc was the fastest way for me to get a stable paying job, the best one I'd ever had, and was frankly probably making as much as my dad had ever made as a postdoc. And so I applied to 2 postdocs where I knew that I would be getting a good mentor out of it as well. And yeah, and then I ultimately chose to move to Arizona to work with Dr. Noah Snyder Mackler in the Center for Evolution and Medicine, because it was pretty close to the biological anthropology and evolutionary genomics I had been doing. But I also knew that if I wanted to stay in genetics, I needed to get hands-on experience with single-cell genomics, which I did not have before my postdoc. And so I was like, okay, well, I guess it's a win-win if I get a good mentor and relevant experience. And Noah was great about letting me be honest about not wanting to be there for more than a few years. Yeah, because I didn't want to get stuck in as much as I wanted to teach, the reality of the current situation was pretty obvious to me, which is there aren't enough jobs in academia. And so I was going to try to get to industry or something alternative to academia. But this was a decent way for me to get a skill set. And it ended up being a very good thing because of the pandemic.
What was the postdoc research ultimately in? What were you looking at?
We worked on building a single-cell molecular atlas of the rhesus macaque brains so that there could be a evolutionarily closer and still, an evolutionary and closer primate brain model for understanding neurological processes and ultimately actually like primate aging as it ends up in the brain. I didn't work on the aging piece, but it's still ongoing in Noah's lab. So a way to look at how primate brains age naturally in a naturally kind of social setting, because these macaques were able to form their own social bonds and social groups on Cayo Santiago. In Puerto Rico.
And you mentioned there sort of the genetics at the single cell level. I know there's definitely a big push now for researchers making these single cell atlases. Can you just give us a explanation of why that single cell exploration is so valuable?
Yeah, well, so traditionally, a lot of what we understand about the brain has been dictated by the amount of detail we can get on it, right? And especially in humans, ethical research does not mean you can get to the cell level. And so a lot of that has been with electrophysiology and MRIs and that kind of functional testing. That's indirect, but as things have moved forward, we started getting bulk RNA-seq from a lot of brain samples. And so that just means taking a whole hunk of tissue with all of the extracellular bits and the intracellular bits, and sequencing that as a whole. And that was good for a while until we were like, well, we can't actually tell what cells are responsible for a lot of this. And so now single-cell genomics is moving towards kind of revealing that complexity at the single-cell level. I'm trying to think of an example. Okay, I'm going to pick my favorite example, which is astrocytes, which are a non-neuron type of cell in the brain that had they had traditionally been thought of as the glue that holds the brain together, because they kind of work in large part to provision metabolites to neurons, because neurons are doing kind of like the electrophysiology stuff for the signaling in the brain, and they don't do a lot of their own metabolism. So in large part, astrocytes can kind of shuttle that stuff to them. But when you're looking at things like Alzheimer's disease, which is not my expertise, but I have written grants on it, so I know something about it. The key to a lot of what is probably, in my bias as a cell biologist perspective, I think a lot of what is going to be key to understanding better the progression and development of things like Alzheimer's disease is understanding individual contributions from different cell types. It is, however, not going to be the only standard for figuring all of that out. What I mean by that is, I think there was a lot of hope that single-cell genomics would reveal everything we needed to know. There's a lot of optimism there. And what I think I'm seeing more of now is a realization that you really need to take a full systems biology kind of approach to these problems, where you combine not just the single cell exploration, but the bulk observation at the tissue and kind of like macro environment level.
So, was data management a large part of your postdoctoral work?
Yeah, because a lot of single cell work is iterative. So you try to figure out the broad strokes of what's happening in your data, and then you have to go back and refine it. And it's this process over and over again. And so it becomes pretty confusing if you're not really diligent about how you're sorting all of your data and all of your attempts to explore that data.
Is that how you then came to Sage Bionetworks then? Through that sort of interest in data?
I found Sage Bionetworks through someone who also works at Sage already, Abby van der Linden, who we both were at UMass Amherst for our PhDs together, and had both expressed interest in alternative to academia careers. And she and I had both been in some of the same kind of settings trying to learn bioinformatics, and she remembered, and she was like, hey, if this is what interests you, check out this place, and then she sent me a specific job listing and was like, I think you'd be really great for the team, you should totally apply if you're interested. And that's how I found Sage.
Gotcha. Do you see yourself as a visual learner? Like, I know now you deal with a lot of data, and I feel like some people might see that dichotomy between sort of data and numbers and then the creative, artistic side. Like, is that a false dichotomy, perhaps?
Oh, that I would say it's definitely a false dichotomy, because I wouldn't even consider myself good at math. And I learned when I was a cashier how bad I am at looking at numbers, because I always flipped numbers. And I'm still a data scientist. So, yeah, I do prefer things in visual format most of the time, but yeah, I would say it's a false dichotomy. I was really also very good at critical reading and in my English courses. I was pretty good at those. I am creative and resourceful, so I usually try to find a way to understand things, even if it's, going and watching videos about how other people understand math. So I just, knew it was something that wasn't my strong suit, and tried to figure it out myself. Tried to be really honest with myself when I wasn't good at something, and how to improve those skills to do the thing that I knew I couldn't be good at.
Sure. I mean, with all the information out there, like you said, and the resourcefulness, I mean, I think that's the important thing. If you have the curiosity, then you can set your mind to things and kind of learn anything you want these days.
Yeah, it's awesome. I only just realized how lucky I was that YouTube got really big at the same time that I was kind of in undergrad. And so I think it really helped, because I would learn from Crash Course Biology videos, and then if I didn't understand something in the midst of being a teaching assistant or teaching my own class, I would just admit that I didn't know, and this person explains it better, so we're gonna watch this.
Yeah, YouTube is amazing. Even though it's been around a long time, I feel like the level of content and the quality is just, I mean, it's studio level stuff now on everything.
Yeah, there was a long time that I wanted to, my goal was to just move to Missoula, Montana, so I could work for Hank Green. And then I realized that it's really a very small company, so I can't put all of my eggs in that basket. So
Yeah, his his empire is amazing. I mean, he's like got the market with all his channels.
Yeah. Well, I don't want to go off on too much of a tangent, but
That's what we're all about here.
Yeah, he and John Green have Don't Forget to Be Awesome as I don't even know what you would want to call it, but it is a system for helping other people make things they're really passionate about. And it was actually something I was going to propose that we try within Sage for a science communication thing, because it seems like a really great resource.
Yeah, those enablers are extremely important. So you're what some people might call a data curator. So people might be familiar with an art or museum curator, but what exactly is a data curator and why are they important?
So a data curator, what we do is we ensure or try to ensure that data produced by one person or one group can be properly utilized by another, in terms of data reuse, but also just in terms of cataloging that data, right? Making sure it just doesn't disappear. And yeah, so that means including as much metadata about how the data was collected as possible.
What is metadata exactly?
This was something I was terrified about when I first got the page. I was like, what do I mean by metadata? Because to me, metadata was just like that information you got about like a picture when you tried to figure out what resolution it was. But metadata is information about the data itself. And so it is confusingly also kind of data. It's going to look like data to people. And so, but in actuality, the data is going to be what you measured, and the metadata is going to be the information about all of the other extra information about that data that came from the measurement. So how you performed the experiment, any variables about what might have gone wrong during the experiment, and all of that kind of information. And I will say with the caveat that I work specifically on these big data genetics omics projects, so I'm coming at it from a pretty narrow sort of perspective. But the data can also be like recordings or images. It doesn't just have to be numerical.
Depends on the data set. Yeah.
Yeah.
It's so funny because when you say that probably to most people, they just think, well, someone uploads data and someone else can use it. So obviously it's a lot more complicated than that. What is that process like? I mean, you mentioned metadata. What else is going into that in terms of the curation, making it usable and findable for people?
Yeah. I think that is probably everyone's first impression about data, and it certainly was mine way back when I started getting into these bigger data sets that weren't just maybe like 24 time points. So the reason we need metadata is because it's kind of the same reason why you still need directions to go to a place you've been like a ton of times, in that your brain doesn't always record all of the information that you need to recreate a thing. And so you have to write it down and be very diligent about all of the information one would need to recreate what you did. And that's how we try to do science from the scientific method perspective. Yeah, so it's important to have as much information about how you got that data as it is about how you analyze that data, because at any given point there could be a variable at play influencing your results that you weren't intending to capture but did anyway. And so my example is at the early days of single cell sequencing, and to some degree, depending on the platform you use, some of the biggest signal people were seeing in data was which day it was sequenced. So batch effects. That's kind of what some people mean when they say batch effects or technical effects in data that show up as an artifact or just something that is not the answer to the question you're trying to ask. And so if you have that information, which you might not think is pertinent, unless you've tried to do this yourself, if you just thought, like, I'm going to upload this to, for instance, the short read archive, and I'm just going to give them what I give them because I want to make my data available. Step one is, yes, it's great that you're making your data available, but people can't use that data if they don't understand all of the nuances that go along with it, because then at the end of the day, when they're trying to make conclusions about the biology, they won't know how to properly control for all of the other things in life that happen, even when you're in a controlled setting like a laboratory.
It's some of that detective work. It's like things you thought might have been trivial at the time actually end up making a big impact and cause the effect that it's seeing.
Absolutely. I have it's not really a great example because at the end of the day, I accidentally threw out the sample, so there wasn't any data. But part of what makes me good at being a data curator is that I also have a lot of experience doing the hands-on bench work to isolate whatever nucleic acid from a sample. And so I know from personal experience just where that can go very wrong in that process. And the funniest / most expensive and hurtful example I can give is when I was learning how to do what we call library preparations, which is creating a sequencing library from a sample. And I've only worked with RNA. And so you essentially have to extract RNA from a sample, turn it into DNA, tag it, and then sequence it. But that process took between 2 and 3 days. At the time I was doing 4 to 8 samples. And so that's a lot of time to then at the very end go and then just like accidentally discard your sample with all of your hard work, which is what I did. And so that's when I learned that I needed to write my own protocols with very explicit checkboxes for me to mark off so that I did not do the thing. And I also created a very angry post-it note for myself to be like, you need to put away, you need to not do this again. Don't hurt yourself like that.
Get it together.
Get it together. It literally said, you have come too far to screw up now. And I saved it and I put it down every time. But yeah, so it's not a great example for why metadata is really important, because there wasn't any data at the end. But when I was talking to my advisor about, well, there's no DNA in this sample. And she was like, so what do you think you did wrong? And then it was, racking my brain to figure out, oh, for 3 days, the sequence of events was A, B, C, until you get to the end where it has to be something kind of like A, C, B. And I was in too much of autopilot so that I still discarded when I should have moved it to a tube for saving. And she was like, there you go. Please don't use the lemur brains. We don't have a lot of them. So yeah, it was learning through my own mistakes.
Yeah, I think we all need those self post-it notes here and there. And it is really interesting that you've had that wet lab experience, and now being on the data side, you can sort of extrapolate to what some of those weak points, maybe bottlenecks are. So that's pretty interesting. Do you also feel like, coming from an artistic background, are there any comparisons between art curation and data curation?
Well, I honestly don't have a ton of experience with art curation in and of itself. So, but I mean, in terms of what I do know about how carefully you need to handle samples, no matter if it's a very, oh no, I have a very bad reference for this, which is, what was that Nicolas Cage movie where he stole the Declaration of Independence?
Oh, National Treasure?
National Treasure.
It's an
Right.
Oscar winner.
But she was, I mean, the leading woman in that was a curator, I believe. And when they were trying to find the secret code on the back, which is probably just how you would have to, they were handling it with a lot of care because it's a very old document, and a lot of art, also old art needs to be handled with care. And so, there, I would guess that there is definitely a little bit of chemistry involved, in preserving art at any rate. But yeah, I guess, do you have any more insight directly into art curation? Because I just unfortunately don't.
No, not really. I mean, it just struck me with the cataloging of the different pieces, and making that available to different galleries, or different people for viewing. And I was just kind of thinking about any similarities there with data users.
Yeah, I mean, I would imagine that both requires a human input on some level, because I know we're going to talk about AI. But I would say it does require human input on some level to really truly understand, or for an art curator, probably walking into a new system, and for a data curator or virtually walking into a new repository, understanding the legacy of why those two things are different and why people chose to set them up differently is going to be really important. Because then you're just going to spend your whole day going, well, why did they choose to camel case this? Right? Why did they choose this version of this word and not this other version? Yeah. And like, I could only imagine trying to give credit where credit is due can be incredibly difficult for art that is so old we don't necessarily know where it came from, right?
Right. Yeah, I mean, who knows? Any art curators listening, maybe they can get in touch and
I'd
set us
love
straight.
to hear it.
And any Linkin Park or Nicolas Cage fans, also get in touch.
Yes.
So you mentioned genomic data sets. And so, yeah, kind of tell us about what kind of data you're managing. Are there certain projects, diseases you work on in terms of looking at those genetic output?
Yeah. So during my time at Sage Bionetworks, I have worked on a number of different projects. I'm going to talk about mostly just the brain related ones because that's my bread and butter. So for about a year, yeah, I'd say solidly about a year, I was the biomedical data manager for Psych Encode. Um, and that is a consortium of like-minded researchers. I think it started about in 2015, well before I was involved. And the goal is to study a variety of different neuropsychiatric disorders and diseases, and in order to better understand them. And so that comes with a lot of very careful data governance in terms of ensuring privacy for the patients who opted into donating their samples. A lot of the data was posthumous, so after the patient had passed away, and so there was a lot from the data governance perspective for that project, and it included and still does include, actually. They just published a huge science or are about to publish a big set of papers from this project in the journal Science. If it's not out already, it will be soon. But that includes disorders like autism and major depressive disorder and schizophrenia, and even PTSD, which I think is a very rare. Samples from folks with PTSD are pretty rare. And so it's going to be a really great resource for studying PTSD, because it is not something that we have a lot of access to, because I don't think we're very good at spotting PTSD in non-human animals. So yeah, and so it was a pleasure to work with researchers working on that field, and being able to come at it from that perspective. So that was the bulk of my time at Sage Bionetworks, and now I'm working more closely with the Alzheimer's disease team on different Alzheimer's disease datasets. They also I'm not directly involved with Treat AD, but it's this really awesome consortium and website, Agora, where folks are trying to approach Sage Bionetworks' mission of decreasing time from bench to bedside by kind of collating a lot of the information that we do know that has a lot of really good chance for success in terms of turning gene targets for interest, for instance, into therapeutics for Alzheimer's disease.
It's amazing that you're seeing that impact of the data sharing and the reuse. And how do you think that's going to continue to shape the field, especially now as we bring in AI tools in? And how are they currently being harnessed? What do you think the future is?
Yeah, I also admit my bias to not fully diving into AI yet. I am a suspicious primate, I guess. But I think it's gonna be great, because so many people are working on it, and that includes so many people who come at this from a data governance perspective. Having people who think about policy and patient rights involved is paramount. And at least from my perspective from Sage Bionetworks, like, we have a whole governance team, and that makes me very optimistic about the future for integrating AI with data reuse and kind of findability and accessibility moving forward. But I do, from my personal experience, I think it is still going to remain a split between automation and human eyes and brains. And just because we can grow brain organoids in a dish does not mean we can recapitulate the human brain in terms of machine learning and AI. And I'm not sure we would want to. I'm sorry, I read a lot of sci-fi as well. But yeah, I think it's moving forward in a good direction, and I have been impressed by the scientific community's ability to kind of like pump the brakes when it seems like it's going a little too fast, in a way that might compromise patient data privacy.
Yeah, you mentioned the governance team there at Sage, and privacy and ethics is something that we take very seriously. So there must be a lot of crosstalk there between the data curation side and the governance team. So when data comes in, are you then engaging them on what controls need to be in place? How does that work?
Yeah, Sage and our team have a good system in place. So anyone who is using our Synapse platform has to agree to use the data in an appropriate way through a click rep. And any of our members who are submitting data through like an NIH funded grant or consortium are expected to also have signed and adhere to our data use and reuse standards. And so that helps us in the first place in that no one is trying to give us patient names, right? Like that's never going to happen. No one who works with us, I think, would miss that big caveat of not giving us protected health information. And so we have to do some checking for that. And so we know kind of the most common mistakes researchers can make in terms of protected health information. And so we know which things to check for deliberately as things come in. But we also work very closely with the governance team. We have folks from the governance team working with us as we work towards curating all of the data we present to the Alzheimer's disease knowledge portal. We work together to ensure that nothing that might compromise any of that trust between clinicians and their patients, or scientists and their the people that they have data on, to make sure that is our first and foremost our primary concern.
Yeah, it's a big push for all the data to be open access, and that's great. But these due diligence controls do need to be in place. So that is great to know.
Yeah, absolutely.
So do you have some advice for people who might want to pursue data curation, bioinformatics? What are the kind of skills or qualities you think someone might need to embody for this kind of career?
Yeah, that's a great question, and it's something that I'm always thinking about because I'm still always kind of like 2 steps away from putting my mentor hat on. And yeah, I think what I would most emphasize for folks going to academia at all and who would like to get into bioinformatics is that you do some soul searching and you know your limits, right? So I'm not a competitive person, and I figured that out in grad school, so a little bit late in life. Not a competitive person. I was not suited for what can become in some really high stakes, highly funded fields, like something like Alzheimer's disease research. I knew that I wasn't cut out for cutthroat grants and getting resources, because my perspective is that everyone should be allowed to do science, just like everyone should be allowed to do art. And so so knowing your limits and knowing yourself well enough to know what is going to keep you happy with your decisions in your career, as your career forks and all of that, knowing truly what you want, or some in the realm of what you want is pretty important, so that you don't lose yourself along the way, because it hurt a lot that I could not reserve any creativity to make visual art when I was in grad school. And if I could do it over again, I would have absolutely made sure that I didn't lose that part, right? And also to if someone suggests you can't do something and they don't think you can do something, but you're sure that you can, go figure out how to make it happen, and just, just some healthy selective ignorance, I think. Of that person said no, but I'm pretty sure I can do it. I'm gonna go try. And don't pay for anything that you can get with for free or for less money, right? So books over expensive courses, free learning tutorials over expensive degrees, right? There's a lot that you can find that is a really great resource for learning a lot of these skills without having to rack up a lot of debt like I did.
What do you feel like you're motivated by then? Is it being a mentor, or is it creativity? What do you think?
I used to think it was just trying to understand diseases better. That was kind of why I went to get a PhD. I got a PhD in molecular and cellular biology, which sounds specific to people outside of the field, but is so incredibly not specific once you're in the field. And so I've realized now that the motivation was only ever to understand myself better, and to be able to give back that knowledge to folks who were not as privileged as I am to be able to take this path.
Nice. It's a noble goal. So
It's also
when you're
very
not,
self-serving, so.
yeah, well, no, it's great. It's great when you can have that balance, right? You get what you want, but you help people along the way.
Mhm.
So when you're not managing genomics and data models, how else are you spending your time? I know you mentioned sci-fi books there.
Yeah, I am getting back into how excited I used to be about sci-fi. So yeah, a lot of reading. Lord of the Rings is my favorite book series ever.
Favorite character?
Eowyn.
Oh,
Yes,
yeah, she's a badass.
she is. Love her. Eowyn and Aragorn, really. And I watch a lot of people playing video games on YouTube and Twitch.
That has become a massive industry. What is going on?
It was what kept me partially sane during quarantine, because I moved to Arizona when I didn't know anyone and then was trapped in a tiny concrete apartment. And so I totally get it. And I'm also not good at most video games, but I like them for the story and the humor. And so that's why I watch a lot of people. And I've also started trying to play my own. Like, I've always played The Sims. I've played way too much House Flipper. And Minecraft. So
I was a big Roller Coaster Tycoon fan, I've got to say. Making those crazy rides, people nearly die.
I was always too empathetic to even create a haunted house in The Sims. Like, I felt too bad killing off Sims. So I would just watch other people who weren't that empathetic to it.
Did you still play around with charcoal and other art?
Yeah, I have bought so much stuff for my own little studio now. Like, I even have an easel at home. And so I'm in the process of planning a lot of art right now. My artistic endeavors have mostly like gone into like tattoos so far. Since I joined Sage and had more brain space to be creative and artistic. But yeah, so I'm in the process of like planning out a bunch of art, and I realized that I've been like anxious about getting started again. And that is just my classic self being afraid I'm not good at something. And so I took an art class at a local community center to kind of get myself back into it. And that was really helpful. And so, yeah, I think I might actually just make some more crow art. They're remaking it. I might as well, I don't know, give some of my family.
Full loop, back where we started.
Everything
I
comes
love
full
it.
circle.
Sometimes it takes that community aspect, doesn't it, to get back in the groove?
Yeah. And as an atheist, I did not fully understand it until for way longer. Because when I was coming into my atheism, that it wasn't really there wasn't really a lot of people that I liked being around that shared the same beliefs. And so yeah, I have such a fondness and reverence for community-based things now that I never realized until I was like very alone during the pandemic. So
yeah, it's such a important aspect of our lives, and actually a big predictor of mortality, it turns out. So it may be good to have a good community around you.
absolutely. And it's also part of us as something that was really profound that I took away from my dissertation, which was mostly on studying how humans have evolved a brain so distinct from other primates, despite being very closely related. A big part of it was sociality, and our emotional intelligence. So a lot of the brain regions that have evolved to be distinct in humans, at least when compared to our closest living relatives, the chimpanzees, are the parts of our brain involved in social aspects. So
Yeah, it's incredible. All right, well, I think we will wrap this up with my final question that I want to throw at people to finish the story arc, which is, if you could give your one piece of advice or wisdom in the area of work, productivity, growth, health, relationships, anything, what do you think it would be and why?
find a way to make yourself interested in the things you have to do. Even if it's making a silly pun, like the only reason I remember what the single letter, I don't need this knowledge anymore necessarily, but the only reason I remember the single letter representation of the amino acid tryptophan is because my advisor taught it to us as tryptophan, and it doesn't matter how silly that sounds. I'm always going to remember it. So I try to take that approach to my work when my brain isn't fully invested in a thing that I want to do. I just know I have to find a way to make myself invested in it.
Sure. Yeah, find joy in it, and the productivity comes along for the ride.
And if it doesn't, maybe you need a nap or a cookie.
Great advice. Well, Trish, I really appreciate you sharing your stories with me today. Where should people go? Maybe if they want to follow you or learn more about your work, are you on LinkedIn, social media?
I am on LinkedIn, so you can find me there.
Great. All right. Thank you so much.
Thank you.